Namaste everyone. This is Dr. Pratibha K.S. Head, Department of Biotechnology at KLE Societies S. Nijingappa College, Rajayanagar, Bengaluru. Today I'm before you to present 6th semester B.Sc. Biotechnology Practical Examination Scheme BTP-602 Planned Biotechnology. Okay, before I begin, I request everybody to kindly subscribe my channel to support. So now go through the practical biotechnology examination scheme and questions. BTP 602 planned biotechnology. Question number 1. Isolate the protoplast or chloroplast from the given sample A and stain. This carries 7 marks. Here, principle and procedure 3 marks, performance 3 marks, result 1 mark. Okay. Then question number 2. Encapsulate the given sample B and prepare synthetic seeds. This carries 4 marks. Performance 2 marks, principal 1 mark, results 1 mark. Okay. Then question number 3. Demonstrate the procedure of surfaceization of explants given. Okay. Or you can also do inoculation of explants. Okay. Inoculation of given explants you can do. Okay. Here this question carries 5 marks. Here explants for this maybe anyone can be given, that is shoot tip or nodal or maybe anther. Okay. Here for this question, examiner may ask you to inoculate shoot tip, nodal or anther, or they may ask you to do only the surface sterilization. Okay, there are two concepts in this question number 3. That is, one is just a surface sterilization. Okay, when you get that particular explant from the garden or outside natural condition, and once you brought to the lab, how are you going to sterilize it? So you can show to the examiner how the surface sterilization can be done of the particular plant culture explants. Other one is inoculation of explants. In front of the examiner in the laminar airflow chamber, you need to show how to inoculate this particular explant to the tissue cultured bottles. Okay. So fourth question is identify and comment on the spotters C, T and E. So this carries 9 marks. In plant biotechnology, spotter carries 9 marks. In industrial biotechnology, only 2 spotters will be there, and that carries 6 marks. Okay. Here, spotters identification 1 mark, comment 2 marks. Okay. Other than this spotters, you need to also answer Viva O's, that carries 5 marks. Then class records 5 marks. Okay. And here you can see the spotters list. I'll be giving that list also. You can go through all these spotters. Okay. For example, you have here growth hormones like IAA, BAP, 2,4-D, kinetin, NAA. Okay. And then in this, any two of the following, we need to keep in the examination any one of the following. Other than these hormones, we can also keep here micro projectile photographs. Okay. And here photographs of callus culture, or you can also keep tubes, then suspension culture. Okay. Then anther culture, then here stages of embryo development, somatic embryogenesis. Okay. Then we can also keep somoclonal variation. All these photographs we are going to keep. You need to identify those spotters and you need to comment on. Okay. So let us go to the question number 1, plant protoplast isolation by mechanical method. Here you need to write the principle and procedure. This carries 3 marks. Performance 3 marks, result 1 mark. So total 7 marks you are going to write, you are going to perform for this experiment. Okay. Then here you can see the plant protoplast isolation mechanical method, aim of the experiment and principle of the experiment. You can go through all these things. I'm not explaining here. Kindly make a note, make your own hard copy of all these questions and answers of the practicals, and that will be easy for you to write in the examination hall. Okay. Then after that, you need to write requirements. Okay, here we are taking forceps, blade, cover slip, slides, 13% mannitol, rio leaves, etc. Okay, then procedure. This is the procedure you need to follow to show to the examiner. Okay. Then this is the result. Compulsory, you must write result. Being a final year, you should not forget writing this result, because many times students will they'll perform the very good experiment. Performance will be really good and they'll show it to the examiner through microscopic view, but they'll forget to write result. Okay. So kindly balance. Don't be very happy also, very enthusiastic also. Balance during the examination hall and try to fulfill all the requirement. Write the result properly. Okay. And underline the result. Okay. And leave that one for the examiner for the valuation purpose. Okay, this is the result you need to write. The next question is question number 2. Preparation of synthetic seeds. Performance 2 mark, then principal 1 mark, results 1 mark, total 4 marks. Okay, here synthetic seeds, same of the experiment, then introduction is given here. I'm not going through all these things. You just make a note of this one. If you are not able to catch, you just pause the video and write all this information. Okay, or you can take screenshot and you can study. Okay, but don't just have a look. You must write all these answers. Okay, and you study only this much information, more than enough for your practical point of view. But for Viva, you need to study all your theory concepts. Next here, the steps involved in making artificial seeds. Okay, I've given here, you can go through this. Steps involved in making artificial seeds. Okay. Hope I'm making myself clear to you. Then here, note, then materials required to perform this experiment, like 2% sodium alginate, groundnut embryos, 0.1 molar ice cold calcium chloride, beakers, pipettes, and petri plates, and many more. If you want, you can keep that with you. Then this is the procedure to perform preparation of synthetic seeds. Have a look. Next, this is the diagrammatic representation of preparation of the synthetic seeds, right? By somatic embryo genesis. This diagram is not mandatory in the examination time, but if time permits, you can write, otherwise it's not mandatory, okay? Then you have to write a discussion about preparation of synthetic seeds. If you miss anywhere now, kindly follow this slide number. Next, what are synthetic seeds? They may ask you in the examination, so you can write. And even in plant biotechnology, if you're not able to, you're not getting properly theory notes, stuff, all that things. No, I have made some other videos, previous videos. Please go through plant biotechnology. Just listen to those videos. That is more than enough, and you can get ready for your Viva questions. Next question is question number 3, that is demonstrate the procedure of surfaceization of explants given, or you can also go for inoculation of given explants, that may be anther, shoot tip, or nodal. Okay, for surface sterilization you may be asked with this anther, shoot tip, or nodal. Okay, here performance carries 5 marks, so total 5 marks. In that you may guess shoot tip, I don't know, shoot tip, nodal, and anther culture. So this is the aim of the experiment of plant propagation through tissue culture. Okay, aim is to raise shoot tip culture from the given plant material. Okay, this is the aim of the experiment, and this is the introduction related with the shoot tip culture. Then significance of shoot tip culture. Then these are the requirements to perform this shoot tip culture. And here you can see the procedure of shoot tip culture. Have a look. If you find very difficult with this procedure, please go through this flowchart procedure of shoot tip culture. In the examination hall, if examiner asks you to write only the procedure, always follow this procedure, okay, by writing in a flow chart. Okay, don't follow this one like a sentence in a bullet points. Just go for writing the procedure or shoot tip inoculation or your surface sterilization, whatever will be there. No, follow this particular flow chart for shoot tip culture, nodal culture, or anther culture. Okay. And before concluding, write that result, whether you have inoculated the explant or whether you have done the surface sterilization of the explant. Based on the examiner requirement, you need to write the result. Then plant propagation through tissue culture, that is nodal culture. Okay, nodal culture, nodal region is the raw material or source for this particular tissue culture experiment here. Aim of the experiment, okay, to raise nodal culture from the given plant material. The introduction part, and this is about the requirements. And this is the procedure. As I told you earlier, take screenshot or pause video and you can make a note of this one. Kindly write all these points in your notebook so that will be easy for you during the examination. And this is the flowchart of nodal culture. You can refer this flowchart for the nodal culture instead of going through our entire procedure. You can keep this in your mind. Then anther culture, aim of the experiment to prepare anther culture. This is the principle and requirements to perform anther culture. Next is the procedure. The last one is here for 6 semester practicals in industrial biotechnology. That is, sorry, this is plant biotechnology. That is the question number 4. That will be spotters. Identify and comment on D, E, and F. You'll be asked 3 spotters here. Here is a mistake. It's not industrial biotechnology. It's plant biotechnology. Okay. Hope you're able to recall this one. Right? This is somatic embryogenesis. Identify, you can write in the examination hall in that paper. So question number 4, spotter, C, D, what is given there over there? D, E, F, right? D, E, F. So spotter D identified as somatic embryogenesis. Then you go back to your place and write the relevant points or comment on this somatic embryogenesis. And wherever you find important or relevant or key points or keyword, kindly underline those. Okay, this is the second spotter here. Okay, what is this? Yes, you are correct. Micro projectile, right? Gene gun method or particle bombardment, you can say. Okay, this is the gene gun method or biolistics method or micro projectile spot. You can write spotter E identified as micro projectile and go back to your place and comment on that particular